Hello, welcome to Biotanemy.
Transcription in prokaryotes. Transcription is the process by which RNA or ribonucleic acid to be synthesized form DNA of deoxyribonucleic acid. The steps of transcription are divided into three. They are initiation, elongation, and termination. 1. Initiation of transcription. The initiation of transcription will begin at particular DNA sites, upstream to the site from which transcription begins, called the promoter. This promoter begins from the sequence 5-TATAAT3-, which is also known as Tata box at the position of 10 elements, or at 35 elements from 5-TTGACA3-. These transcription start sites are recognized by an enzyme called prokaryotic Rna Polymerase. The RNA polymerase comprises core polymerase, which consists of alpha, beta, beta-, and omega subunits. After the sigma subunit will bind to the core enzyme to form a holoenzyme. The sigma subunit will help the RNA polymerase to recognize promoter sequences in the DNA. The binding of RNA polymerase to the promoter sequence leads to the formation of the closed complex. Then the RNA polymerase will separate 10 to 14 bases of DNA strands, starting from -11 to +3, through a process called melting. The opening of DNA will lead to the formation of an open complex. This process is called as isomerization, and the synthesis of RNA more than 10 base pairs long will occur to start the DNA synthesis. The sigma subunit will exit from the complex to form the tertiary complex. 2. Elongation. The dissociation of sigma subunit will allow the core RNA polymerase to move along the DNA template strand and synthesize mRNA in the 5-to 3- direction. 3. Termination. The termination of transcription will occur through two different mechanisms. The first one is Rho-independent, and the second one is Rho-dependent. Rho-independent mechanism. In the Rho-independent mechanism, the transcription will be terminated because of specific sequences which begin with CG island followed by some random sequences and again CG island to form a hairpin loop, and finally a continuous repeats of AAA nucleotides. In the Rho-dependent mechanism, the transcription will be terminated with the help of a ring-shaped single-strand binding ADPase protein called Rho. The Rho protein will bind to the single-stranded RNA as soon as it exits from the polymerase enzyme complex, and will unwind the DNA-RNA hybrid when it encounters the terminator sequence.
Thank you. Please like, subscribe and share. See you again in a new topic.